Could mobilizing more stem cells into the blood actually reduce your reservoir in the bone marrow?
It doesn't. And I know that it's not something that is easy kind of wrap our minds around it, but I think the best analogy will be like a well. You know, a well, you have like the water vein, and the more water you take, the more water you have. Like you won't have your well is not going to go down. If anything, your well will provide more. I'm not saying your bone marrow will provide more, but what I'm saying here is that while your red marrow shrinks, the red marrow that you have at any point in life just produces stem cells endlessly. It's an endless reservoir of cells. Stem cells in the bone marrow have telomerase, so telomeres do not shorten. So these are immortal cells. So when you release them, they multiply. Like the amount of stem cells, let me put it this way. They did a study to ask the question, when we inject stem cells after a cancer treatment, they normally inject 200,000 stem cells per kilogram of body weight. So, but the question is, when you extract these stem cells from a donor to inject in a recipient, you don't know of these cells how many are really stem cells, because you cannot use antibodies and different things. You change those cells. So you try to do your best to extract stem cells. You don't know how many. So the question was, what if I inject only one? If I'm sure that it's a stem cells, but I inject only one. And the study showed that if you do myeloablation, inject one stem cells, in 6 weeks, you've reconstituted the bone marrow in the whole blood system.
Wow.
So if you have 100 million in your bone marrow and you release 10 million, the 90 million that are left won't have a problem to maintain your bone marrow. So when you release more, you don't have fewer stem cells in circulation.
Well, that's very comforting to know. Yeah, so there's almost like the body has a homeostasis. So
Correct.
it reached a new homeostasis, no matter, even if it's a small amount of reservoir, it will maintain that reservoir.
Correct.
But it is a smaller amount, so you're going to release less. So you want
Correct.
to allow it to.
Exactly.
Okay. I wonder if that stimulates the cells, by allowing them to release more.
It could. This is all the kind of like basic research that I would love to do. So as we get more and more funds to do research, these are all the things to look at. And other things to look at that we cannot say a whole lot about because there's no research, because there's no drug known to do it. Therefore, there's nobody has really invested in that research. It's stem cell migration. When you put them in the bloodstream, they're not doing anything in your bloodstream. They need to migrate in your tissue. That process of migration is really not well documented. So we focus on mobilization, but I believe that migration is also a very important phenomenon. And I'm saying this because as I'm looking at all these plants, when I studied medicinal mushroom, goji berry, and a lot of those polysaccharide type active compounds, after about 30 minutes after consumption, what we saw is a sudden drop in the number of stem cells in circulation. Our first conclusion is that that's it. Our machine is broken, something is wrong, so let's clean the machine, change all of our chemical agents, clean up everything, redo it, and we got the same data. And after a few months, we realized that looks like it's real. And as we did a little bit more research, we found that the receptor, CXCR4 receptor, that is responding to SDF1 released by injured tissues, that receptor increases, the number of them on the membrane increases significantly after taking these polysaccharides. So that means each stem cell has a greater probability, more chance to respond to a SOS signal from tissues. So as they migrate, then they disappear from the blood. So when we start to document some of those, that's when I start to combine them. Because as we mobilize them in the blood circulation, they need to go into the tissue to really be effective. So if I stumble on plants that do that, then I'm including them. But scientifically, there's very little that I can say because it's not very well documented in the scientific literature. When you publish, you need to discuss your data with other data. So in lieu of having that kind of literature, that means I then need to go into more basic research to document the process of migration. It's a lot of money. But
Yeah.
I would love to do it. We will do it one day.